A warm good morning to one and all. We are pleased to welcome you all for the two-day National Biotechnology E-Conclave, HyGene, the challenge of the change, with the theme of emerging biotechnology in the current pandemic. The main objective of the program is to emphasize the role of biotechnology in the current pandemic, and we are also aiming to discuss the career opportunities in the life science stream. Yesterday was the first day of National Biotechnology E-Conclave, in which we had conducted a webinar on COVID detection technologies, challenges in the pandemic world, and a research poster presentation covering major areas of life science. Today being the second day, we had included a bioinformatics workshop and career guidance in life science. Each section will be handled by eminent personalities in the field of biotechnology and bioinformatics. For enlightening the young minds, we also organized various competitions for school and college students. We are honored to have you all as a part of this event. Next, I welcome Ms. Fatima Zahra for introducing our guest speaker. Fatima Zahra, please.
Good morning, all. Hope you all are fine. Here we are in the second day of the event, Hygiene, the Challenge of the Change. And we commence the day with a bioinformatics workshop on sequence analysis by Professor Dr. T. Ashok Kumar. He is the former head of Department of Bioinformatics, Noorul College of Arts and Science, Kumaracoil. He took his PhD from Manonmaniyam Sundarnar University, Tirunelveli, Tamil Nadu, and designated as the former UGPG board chairman at the same university. He has done his specialization in R programming, biostatistics from Johns Hopkins University, Baltimore, US, and completed postgraduate diploma in computer applications from Cybersport Institutes, Kuriturai, Tamil Nadu, and MPhil in bioinformatics from Bharathidasan University. He received honor as academic associate for the program Cloud Infrastructure and Services from EMC Square. He is the developer of bioinformatics blog site BioGem, which is aimed to share inventions, innovations, and trends in bioinformatics. He developed phytochemical and drug target database, online tools like CFSSP server for secondary structure prediction, JMOL PDB viewer for protein structure visualization, BP blast, log blast, and many more. I have immense pleasure to say that we have got an experienced person in the field of bioinformatics to conduct this workshop. And I am here to welcome Dr. Ashok Kumar, sir, to this wonderful session. We welcome you, sir, and thank you for joining us. Over to you, sir.
Thank you, Fatima Sahara. Good morning, dear participants and faculty members of Kerala Agriculture University. I am happy to present workshop on sequence analysis. My name is Dr. T. Ashok Kumar. Can you able to hear my voice?
Yes, sir, you're audible.
Okay. Okay, once again, good morning. Let's start the session. Today we are going to discuss about the following in the following area. There is a sequence retrieval from the database, pairwise sequence alignment and dot plot, sequence search from the database by alignment, multiple sequence alignment, and phylogeny. Then the last one is physiochemical properties prediction. Before entering into workshop session, I will give some introduction about the sequence and the sequence analysis. Throughout this session, I will be discussing about sequence analysis based upon the bioinformatics or computation biology approach. Then, first thing is, what is a sequence? Sequence is arrangement of ordered succession of characters drawn from a finite set called alphabets. This is a computational term we usually use for defining a set of characters. In bioinformatics, we use the term for defining DNA alphabet, RNA alphabet, and amino acid alphabet. Alphabet means a set of defined characters. I think all of you know about that. In a DNA, there are basically A, T, C, G. Apart from this basic, there are also some variants like B, D, H, K, M, N, R, S, V, W, and why, I will explain later. These are all known as a non-standard residues. In a computation aspect, we consider only the letters in the place of a sequence as a residues. Then similarly, for the amino acid alphabet, here as I mentioned, these highlighted blue letters are for 20 amino acid, which are a standard residues, and in the red I mentioned B, J, O, U, X, etc. These are non-standard residues. In bioinformatics, basically we consider sequences. So there are a lot of issues while comparing sequence. The thing is, if you see here in a DNA, there is a letter A, also in the RNA, also in the amino acid. This is some confusions while we searching through the database, because it is matching in the 3 alphabets. Also similarly, T is also matching in the amino acid, C is also matching in the amino acid, G, like that. The alphabets are repeated. So usually in the either a database or in a sequence analysis, they define every alphabet as commonly a biological sequence alphabets. In olden database and tools, they only follow the standard residues. But nowadays there are so many variants, so they now consider all the variants also. That is the standard and non-standard residues. Okay. Now we have to understand what is the analysis about a sequence. Analysis means we are deriving the properties from a sequence. Basically, sequence is nothing arrangement of these characters known as alphabets. From these alphabets, we derive the features, structure, function, evolution, and so on. Then, in bioinformatics, these analyses are done by comparing the sequence with any database or data set, and also using the tools, we predict the functions. It is usually done through a statistical approach by defining a set of rules, which is also known as array or data set. Okay, these are the standard 1-letter code for the nucleotide and similarly for the amino acids. Just for your understanding, I have given this. Here I have highlighted some letters in red. These are non-standard residues. Similarly, here also I mentioned for the 3-letter codes and 1-letter code for the amino acid. Basically, we consider only the 1-letter code. Okay, now we have to know what is the sequence file format, because throughout the bioinformatics, for example, from basic analysis to drug discovery, we follow a set of rules known as a file format. For a sequence, we use the standard sequence file formats, which are known as a raw or plain format. It is simply a file without any rules. Then next one is a FASTA. This is another type of a standard file format which are used to represent a sequence. The FASTA file format is introduced by EMBL, that is belongs to the EBI. Then other standard file formats are FASTA-Q, GCG, PIR, GenBank, and so on. These are file formats are introduced by the databases. GCG is one of the popular package which was used for sequence analysis, which is somewhat similar to the emboss package. Now it is outdated. Then PIR belongs to the PIR database. GenBank file format belongs to the GenBank. And EMBL file format belongs to the EMBL. These are the most commonly used file formats. But for only sequence analysis, we either use this raw file format or FASTA file format. Basically, FASTA file format is similar to the raw file format. The only difference is in the first line, we can notice that there is a greater symbol followed by one line. This is used as an identifier of a sequence. If there are more number of sequences, after breaking this sequence, the next line again begins with this greater than symbol. So we can able to easily understand if there are more than one sequences. So in the FASTA file format, we use the greater than symbol to break each and every characters and sequence. Actually, in the first line, the greater symbol followed by a keyword separated by either a space or comma, or in some cases there is a pipeline symbol. These are symbols are used to separate the keywords. For example, the first one np underscore etc. This is the accession number. The next one is can you hear? The next one is a type of a sequence and the description and so on. This is first just for identification. The next line is a sequence. Then what is a database? It is a computational term, not a bioinformatics term. Basically, database is a collection of structured information. That is an important point. Without a structured information is just like a raw file. In the database, especially for the biological or chemical database, they form a standard format to classify the data. For example, like a GenBank file format or EMBL file format, like that. Each and every database follow a standard rule to represent the data. So, for example, if it is a GenBank database, they use a standard format to store the data, which is not accessible as a GenBank file format. So, if the information is related to biological content, then it is known as a biological database. Basically, a storage unit is simply known as a database. So now, what is a biological database? Basically, a biological database is categorized into sequence database, structure database, and functional database. Then there is another alternative method used to represent this biological database, that are primary and secondary. Usually it is categorized into primary and secondary. In some cases, there is also another one category, composite or specialized database. The thing is, in the first type, we simply categorize any database based upon the sequence only. That is, as I've already mentioned, based upon the DNA alphabet characters only, then it is known as a sequence database. Then structured database consists of atomic coordinates of molecules. Similarly, the functional database consists of a network, like interaction between a protein to protein, or protein to nucleotide, or protein to metabolic pathways, like that. That is somewhat related to the sequence, structure, and additional data like functions. So such kinds of repository is known as a functional database. But in the second case, basically these all sequence or structured database comes under a primary database. That means purely experimental data. Here in the first case, sequence and structure is a experimental data. The third one is derived from sequence or structure. Like that, in the secondary database, it either consists of a primary as well as a secondary, or purely secondary. The thing is, in the primary, it consists of experimental data. In secondary, they interpret the primary data and add features to the primary data. So it is known as the secondary level. The third level, that is a composite. Composite means combined. It consists both primary, secondary, and additional features. Okay, these are the two types of a classification of a biological database. There are many biological database, more than 300 databases. So for workshop program, I have screened only most popular databases. For nucleotide sequence database, NCBI GenBank is the most popular. It is internationally standard database used for nucleotide sequence retrievals. Then another one popular database is the EMBL. Previously it is known as a EMBL. Now it is also known as a ENA. I have given the link of both. Basically, it is from the same site, eba.ac.uk. Similarly, in the second website, you can see that ENA, that is also belong to the EBI. It is a new format derived from the EMBL. It is standardized format. Then the third popular database is a DDBJ. But most of the data also included in the GenBank or EMBL. Then the next type, protein sequence database. There are only limited number of protein databases. So most of the databases are now combined and known by a universal protein database, simply UniProt, that belongs to the SIS Institute. I will explain later. Then next popular database is PIR. Even though it is popular, the most of the database are retrieved from the UniProt. I will explain it later. So these are the two types of classification: nucleated sequence database and protein databases. Only for a sequence. Now, now our practical section starts. Now we are going to retrieve a sequence from a GenBank. Basically, it is a GenBank database. Even though it is a GenBank, it also consists of a protein database, protein sequences. The thing is, basically, it is a nucleotide. In the GenBank, they translate the nucleated sequences to a protein. So the such formats are known as a GenPept. But basically, it's a GenBank file format. Then these are the two URLs you can directly access from the home page, www.ncbi.nlm.nih.gov, or through this second one. Basically both are same URL. Then about the NCBI, it is a part of NLM and NIH. So now the practical section starts. First you have to open the web browser and enter the URL www.ncbi.nlm.nih.gov. Okay. The next step, we have to select the data. By default, it will direct to the NCBI literature. So for sequence retrieval, you have to select either a protein or nucleotide. In this tutorial, I have selected protein. So we have to enter the keyword according to the protein. Now, in this text box, you can either enter three types of data, either a keyword, accession number, or genome ID, or author's name. That is option to you. In this, I have used a keyword insulin. As I have already selected a protein, I have entered keyword accordingly. Okay. Then, fourth step is we have to click the search button. Okay. Now after clicking the search button, it will redirect to the result page. This is the result page. In this, it will show number of hits matching to our keyword. You can see that the insulin keyword is highlighted in each and every hits. So these are the matching hits. So for downloading the sequence, you have to click the checkbox. If you have to download only one sequence, then you have to click the checkbox. Here I have selected 5 sequence. We can select maximum number of sequences. Actually, in this hit, there are 1,13,427 matching sequences. We can also select all sequences. For our convenience, I have selected only 5 sequences that I have highlighted here. Then the next step is you have to select the send to. There in the right side there is one drop down arrow. If you click this, it will expand like this. Then from this you have to select the file. Okay. Next step, we have to choose the format. By default, it will be a GenBank file format. For our data analysis purpose, I have chosen the FASTA. The next one, sort by, that is optional. Whether you have to sort the sequence according to the this keyword or gene ID or accession number and so on. Here this is the accession number NP_0011 etc. and genome ID 325 etc. So if you want to sort the sequence accordingly, you can choose it. Otherwise, just leave it. Then click the create file button. Now automatically it will save the sequence file in a FASTA file format. Usually the FASTA file formats are saved with the extension .FASTA or .fas or .fa. These are the extension. You must be careful while saving a sequence file in this file format. Otherwise, the tools will not recognize. Okay, that's all the procedure for the GenBank. Then the similar approach is for the EMBL also. Basically, EMBL is now merged to a ENA. So you can directly download the sequence from ENA, or you also go through from the EBA site also. The steps are somewhat similar. The only thing is the format are now converted to ENA file format. In the previous versions, the file formats are not by EMBL. Now the format is a ENA. That's all the difference, but the contents are same. Then about the EMBL, it is a part of the EBI. Now it is standardized and categorized into a new database ENA, European Nucleotide Archive. Okay, now again the practical section starts. The procedure is somewhat similar. First, we have to open the browser, then enter www.ebi.ac.uk. Next step, we have to enter either a keyword or accessor number or author's name, that is similar to the GenBank. Then third step, you have to choose the data. For example, for a nucleotide, you have to select a nucleotide sequences. Then for a protein sequences. I have here choose nucleotide. Then click the search button. Okay. After clicking the search button, it will redirect to the result page like this. There are around 48,65,362 hits. That is not only sequences, some literatures or functionals and genome data, etc., etc. So for a sequence retrieval, we have to click this link sequence that is present the left side. Then after clicking this link, it will redirect to the next page. Okay. In this, we have to select the sequences which are going to download. Actually, the steps are somewhat similar to the GenBank. We cannot able to download any number of sequences at a time. In the GenBank, we have seen that if you select the checkbox and click the option to save, it will automatically save all sequences to a standard FASTA file format, but it is not possible in the EMBL. We have to manually download each and every sequences. So the first step, we have to click the decide sequence that is highlighted in the hyperlink. Then it will redirect to the ENA. That's what I have already explained, that basically now EMBL is converted to a ENA. So you can directly access from a ENA or from EMBL. Both are same. Actually, the ENA consists of a standardized contents of a EMBL. Now, for downloading the sequence, we have to select the FASTA link that is in the right side of this box. Okay. While we click, it will automatically download the sequence into FASTA file format to your system. The sequence is very large because it's a DNA sequence. I have given only a portion of the sequence. Then the next practical is a UniProt KB. Previously, it is known as a UniProt. The meaning is Universal Protein Knowledge Base. They use the term knowledge base because it contains a lot of information apart from the sequence. Like other database, not only it consists of only sequence or features, it is also interlinked to the structural database, functional database, or some other database related to the proteins. So only they name it as a knowledge base. Then it is previously it is a part of the Sysprot. So it is somewhat sometimes it is also known by Uniprot KV/Sysprot because it is a part of a Sysprot. Now EMBL and GenBank are collaborated with this Uniprot. So the data which is present in the GenBank or EMBL is also now available in the UniProt database. So only now it is known as a universal protein database. The thing is, basically UniProt consists of a protein sequence which are deposited by the Sysprot. But due to this popularity, now EMBL database, also known as a TR-EMBL, translated EMBL. The thing is, the nucleotide sequences are translated to protein and deposited as a protein sequence. Also, in about the GenBank, the nucleotide sequence are translated to a protein sequence and deposited to the UniProt. It is sometimes known as a GenPept. Okay. So we can access any database through the UniProt. So only it is known as a universal protein database. So we can notice that the left side they have mentioned UniProt consists of a large collection of a Sysprot that is manually annotated and reviewed. Similarly, the TR-EMBL, translated EMBL, consists of automatically annotated and not reviewed. The thing is, these contents are not experimental data. It is translated, that is curated. Okay, now the practical section starts. Again, open the browser, enter the URL uniprot.org, then enter the keyword or accessor number or author's name in the text box, then click the search button. This is the result page. In this, the layout will be somewhat similar to the GenBank. Here you can select any number of sequences as I have highlighted here. Then there is also another option. You can download entire matching sequence. The file size will be very large. So for your convenience, I have again selected only 5 sequences. Next, you have to choose the format. The default format is the FASTA. Then, by default, the file will be in a compressed format. I have chosen uncompressed format, so it will save in a FASTA file format. If you have low internet connection, then you can choose the compressed. The compressed file format will download the FASTA files in a compressed format, usually in the .gz format. The file size will be very small. Nearly it will take only 5% of data for downloading. It is up to your option. I have chosen uncompressed. Then click the go button. The difference is, as we have seen in the previous databases, we can directly download to a FASTA file format, but in this case, it will display in your browser. You have to manually copy the contents and paste in any text editor, and you have to save to a FASTA file format, that is a .FASTA or .fas or .fa. Another important thing, while we are saving the FASTA files, we have to choose only a standard text editor like a Notepad or some other ASCII editor. You must not choose the rich text editor like a MS Word or etc. If you save the data in a rich text editor, most of the sequence analysis tool will not recognize the file format, because even though if you save the content in the file format, that is in the FASTA file format, it will recognize as only a document file format. So you must have to remember that while saving a FASTA file, you have to choose only standard text editor like a notepad, and we have to save with the extension dot fas or fasta or fa. Okay. uh, Anybody have doubts? Anybody? Up to this, we have discussed about a sequence downloading. That are the basic things we have to understand. But in the sequence alignment, it's strictly based upon the algorithm. So only I am asking, is there any doubts? You can clarify. Okay. The next core area is a sequence alignment. This is basically used for a comparison of a sequence also at a high end for a next generation sequencing also. The thing is, the sequence alignment means arranging the characters of both sequence to uh, form a similarity. For the similarity, we use set of rules for matching the characters. Usually we match the characters at a column wise. For example, here we can notice that there are two sequences. The first one, the first character is also same as the second character, first character in the second sequence. So we consider as a match. Match means same. So in the sequence similarity for matching, we use a column wise comparison. For example, it is a multiple sequence. Again, we use a column-wise comparison throughout the entire sequences. The common rules are, if both characters are same, then we consider as a matches. For a sequence alignment, we only expect matches, but there may be possibilities of mismatches like this AC. Also, in some cases, for aligning the sequence, we use some gaps. It is represented by hyphen. Okay, these are the three rules we use to align a sequence: match, mismatch, and gap. So, for algorithm purpose, we use some scoring schemes known as the match score, mismatch score, or gap score. Sometimes gap score also known as a gap penalty. Strictly, it is a penalty. Penalty means it is like a error. We cannot use a gap. In some cases, there may be no possibilities to align a sequence. So, in such cases, we insert a gap. If we insert a gap, the penalty must be high. The scoring scheme is usually in this order. The match score will be high, and mismatch score will be less compared to the match, and the gap penalty score will be much more lesser compared to the match, or lesser compared to the mismatch. In some cases, the gap score will be strictly negative value. And for a mismatch, we use alternative method, also known as the accepted mutation. That is, we consider the mismatches as mutations. So, there is one assumption that accepted mutation means if there are set of sequences that belongs to the same family. For example, grandparent, parent, child, grandchild, grandchild, and so on. These are all belongs to the same families. So in such cases, if there are some character changes like mismatches, we can also accept that changes. So we know such kind of mismatches as an accepted mutation. So for the scoring, we also use positive scores for the mismatch, but gap penalty are strictly negative values because it is not we expect. The another important thing is, while comparing sequences, we take one sequence as a standard sequence to compare with the other, usually known as a query. The first one will be a query sequence. Then the next one will be a subject, the sequence which we are going to compare with. Okay. Here, if there is a gap in the query sequence, then we strictly term it as a insertion. Also, if there is a gap in the subject sequence, then we known as a deletion. So insertion and deletion, in the short we know, term it as a indel, insertion and deletion. The thing is, if you go for sequence similarity, we expect matches only. In some cases, we may have to insert a gap or mismatches. So it is always not possible to insert a gap in a sequence, that is in a query sequence. But in some cases, there may be possibilities of some errors in the database. So we use the gap in a subject sequence as a deletion. It may be possible chances of mutations, or it may be deleted, or some other experimental errors, and etc. So strictly in the query sequence, we mention a gap as an insertion. We are only inserting a gap to make matches. If you are inserting a gap in the subject, then we mean as a deletion. Okay. So strictly for scoring purpose, we take this as a penalties, known as a gap penalty or indel, insertion or deletion. These are the basic rules we follow for the sequence alignment. Now, what are the types of a sequence alignment we follow to compare two or more sequences? The first type is pairwise. Pairwise means two. We are aligning two sequences. Then second type, multiple sequence alignment. Multiple sequence alignment means three or more. We have at least three sequences to compare, because without three we cannot can't able to differentiate. If there are three sequences, then we compare as a AB, BC, and CA. So for the multiple sequence alignment, we need at least three sequences. Then in the pairwise alignments, there are two types of approach we use to match the sequences. First one is a global sequence alignment. Then second type is the local sequence alignment. Global sequence alignment means the alignment is global, means it compares the maximum possible chances. It will compare from the beginning to end of the sequence. So such kind of alignment is a global. For structural prediction or functional prediction, we usually prefer second one, that is a local sequence alignment, because it align only matching characters, that is the exact matches. The difference between a global sequence alignment and local sequence alignment is, in a local we allow only matches. So we either ignore the mismatches or gap penalty, or we also consider the mismatches as accepted mutation, but gaps are strictly reduced. There are also certain limitation. For example, there is a one gap, they will allow with small penalty. If the penalty is more, the gaps are considered as a separate sequence. For example, I use a match score as a 1, then mismatch score as a 0, and gap penalty as a -2. Then if it is a more gaps, for example, there are only 4 characters and the gap is 4, so it means there is no alignment. So for the local alignment, we strictly reduce the gaps. If there is more number of gaps, we break into subsequence. Okay. Then about multiple sequence, I will explain later elaborately. Then we shall see what are the approaches we use for the pairwise alignment. Usually for the programs or tools, we use a dynamic programming. They use a program to compare each and every characters between two sequences. It is very time consuming. So now new methods are introduced known as the word method. That is, instead of matching with only one characters, we use limited number of characters. For example, one word as a GTC, then this word will compare with the GTC. Here there is only one matches, so it will begin with the GTC with the GTC. Such kind of comparison is known as the word method. It will use limited number of characters as a words to compare with the second sequence. So if you use the word method, it will be less time consuming and result will be more accurate compared to the dynamic programming, and results will be optimized. Now most of the sequence analysis method, even for the NGS also, we use this word method, because if you compare with the billions of sequences, this dynamic programming will be very time consuming. It may take many years. But word method use certain assumptions to filter the large size of a sequences to limited number of sequences, like comparing by the families, then comparing based upon the scores, and based upon the word size limit, and so on. Such kind of assumptions are known as a heuristic approach. Heuristic approach means with a set of assumptions. But in case of a dynamic programming, it is strictly based upon a protocol. So modern computation methods only follow word method. Then the last one, it is a dot plot. It is used to just analyze the sequence. I will explain later. Then the tools used for global sequence and local sequence, there are many tools are used. I have given only the most popular tools, that is BLAST. I think most of you are familiar about this BLAST. Then another one tool is the FASTA. This belongs to the EBI. These NCBI consists of many tools from basic sequence alignments to genome analysis. Then for the multiple sequence alignment, there is one popular tool, Cobalt. I will explain later. Similarly, there is one popular tool, Muscle, that belongs to the EBI. These tools are more or less competitive. Accuracy is more or less equal. Then this is a dot plot, also known as a dot matrix. It is a 2D graphical representation to analyze a sequence. We can able to analyze what are the regions which are matching with the sequences. But for a sequence analysis, we have to choose an alignment approach. In this graph, I have mentioned the x-axis in the top. In some cases, the x-axis will be in the bottom. The thing is, it will compare x versus y. For example, this is a x, then horizontal as a y. We compare each and every characters between sequence 1 and sequence 2. If it is continuously matching, then you will get a perfect match, slanting line like this, because in the both the sequence the characters are matching. So by plotting a dot for each and every matches, you will get like a perfect slanting line in this order. So this dot plot is very much helpful to analyze the sequence. Then there are many types of plots. In the next case, there may be possibilities of motifs. Motifs means repeated sequences. So through this second approach, we can able to find how much number of repetitions, sometimes known as duplications. Here we can see 3 lines, which is similar to the first one. The only thing is there are 2 lines extra, here one and here one. So the thing is, there are some subsequences are matching, maybe here or here, like that. So this second graph will be helpful to find out the number of repeats. Then third type, palindrome. Palindrome, I think all of you are familiar about that. If you take the sequence in reverse also, it will be same. So through this dot plot, we will get like this cross line. So we can easily understand that it is a palindrome. It is usually used for DNA sequencing to compare with the reverse complementary sequences. Then this is partially palindrome, means somewhat similar to the second graph. The thing is, even if it is a reverse, there will be some matches. So it is known as a partially palindrome. Then the fifth and sixth graph, it is known as a micro satellites, then macro satellites, then there is also another type, mini satellites. That means there are many number of short fragments which are frequently matching. You can see here many fragments are repeatedly matching, so it will form like a block. Okay, so these regions are known as a satellites. Here it is very closer, so we say as a micro. Then there is also a mini and a macro. That means the regions are very closer. Then the third one, it is known as a homology. Homologous means in some sequences, for example, uh, it may be belongs to the same families or related families. For example, Homo sapien with the Drosophila or some related sequences. Actually, the species are different, but sequence are similar. Such kinds of sequence we known by homologous. So through this graph, we can able to find out the homologous sequences. Then the last one, actually, it is somewhat similar to this graph, but the one thing we can notice that the line is little deviated. Actually, it must end here in the corner, but it has end here. So the thing is, there is a lot of gaps. Even though there are many matches, there are a lot of gaps. That is insertions and deletions. So using this type of graph, we can able to find there are a lot of number of gaps, simply known as the indel. Okay. Now the practical section starts. First, we are going to see about the pairwise sequence alignment. That is a global sequence alignment. For this also, we use the BLAST. BLAST has a many number of tools. From this BLAST, we are going to choose the pairwise alignment. Pairwise, especially the global sequence alignment. For this, they use a Needleman and Wunsch algorithm. It is discovered by two famous scientists known as Saul B. Needleman and Christian D. Wunsch. It was first introduced during year 1970. After this success, they have discovered another algorithm that is known as Smith Waterman. I will explain later. The principles are mostly similar to this Needleman-Wunsch algorithm. They have made some modifications to improve accuracy. Again, the practical session. Open the browser. Enter the URL blast.ncbi.nlm.nih.gov. Then select the global align. Then it will direct to the next page. Here we have to select nucleotide or protein according to your query sequence. The next step, we have to enter your query sequence. The acceptable formats are either you can enter the accession number, gene ID, or standard FASTA sequence. In this, I have choose the FASTA. Here you can notice that in the FASTA format there is a genome ID. If you know about the sequence, there is no need to paste the FASTA sequence. Instead, you can just enter this ID itself, 6693, etc. This itself enough. Otherwise, you can also enter this GenBank ID, AAF2, etc. Okay. I have just given for your understanding. If you are creating your own sequence, you can just copy and paste it. Then, if you store the sequence in a file, you can choose second option. I have pasted the sequence in the text box, so I have ignored this. If you have a sequence in a file, then you must have to choose this one. Choose file. Then next step, again like query sequence, we have to paste the subject sequence. Then click the blast button. Okay. By default, it will show the description. That is like a scoring scheme they used, course, sequence description, subject description, sequence length, and so on. I have clicked the third tab to show the alignment. Okay, there are totally 4 tabs. First one, description, that is about the sequences. Then the second tab, it will be graphic summary, also known as the color keys. The sequence alignments is represented by color key, but for the parallel alignment, there is no need to choose this graphical summary because only we have given 2 sequence. There is no need to show the summary. I have choose alignment. Here, in this alignment format, there will be 2 rows. The first row represents the scores that is calculated using the BLAST algorithm. Okay. Here, NW score represent Needleman Wunsch score. Actually, for a BLAST, they use a BLAST algorithm. That is a heuristic algorithm. But we have chosen the BLAST program for Needleman Wunsch algorithm. So the score is represented as NW score. But basically for a BLAST, the score will be a bit score. I will explain later. Then next one, identities. Identities means exact matches. It is calculated by percentage. Total number of a sequence by number of identities. Then the next one, positive. It includes both identities and matches. Then the last one, intel score, that is a gaps. Number of gaps that are inserted in the query or subject. That is 56 gaps. Okay. Now the interpretation. Here in the BLAST result, there will be 3 lines. Query, then next one, the symbol representation, which shows match, mismatch, gaps, and etc. Then the third one is a subject row. For comparison, we have to read in the column wise. For example, in the first column, there's a M, M, M. So in the intermediate, in the second line, there is a symbol used as a, the letter used as a M. That means the both characters are same. In older version, instead of letters, there will be a symbol representation like a pipeline. Okay. Either they use a pipeline symbol or the same letter, or in some alignment they use a double dot, a single dot, and so on. In the new format, they use the same character to represent it as a matches. Then here we can notice that a plus that represent the positives. That is positives means that is somewhat like a mismatch, which is accepted mutation. Okay. In these alignments, we have to note that mismatches also consists of accepted mutation. So in such case, they use the symbol plus. If there is no plus or there is no representation, then it is strictly mismatch. But in case there they used a plus symbol, even though it is a mismatch, it is accepted a mutation. That is the meaning. Then there are gaps in the subject. Okay. The one plus point is here there is no gaps in the queries. Sorry, there is gaps. So we cannot accept as a more matches. We must note that the gaps must be less compared to the subject. Okay, if there are more gaps compared to the subject, then the alignment is very poor. Not due to the algorithm, that is due to your sequence. The sequence may be entirely different. Actually, I have choose here one Homo sapien. That is one species. Then another one mus musculus. So it is totally different. If I have given same sequence or sequences belong to the same species, then the gap will be less. But in this case, the two sequences are entirely different species. So there are lot of gaps in this subject. But always you have to remember that gaps in sequence must be less compared to the gap in the subject. Okay, this is a new format. In the olden formats, there are well some other scores. Okay, you don't worry about that. Okay, then fourth tab that displays the dot plot. As I've already explained, here the first sequence, that is our query is represented in horizontal in the bottom. Okay, in some cases it will be represented in the top also. So accordingly you have to interpret. In this, in the top it is the first sequence, and in the horizontal it's the second sequence. But in this case, in the bottom, it is the first sequence, and horizontal, that is a y-axis, it is the subject sequence. Okay, you can notice that it ends at the corner. So it is homologous. That is the dot plot interpretation. Okay, this is a homologous. Then, okay. Now next practical, that is a local sequence alignment. Actually, it is similar to the global sequence alignment. The only changes is in this local sequence alignment, they have reduced, the authors have done small changes. That is, they strictly reduced the gaps. So there is no possibilities of multiple matches. Then the second change is in this mismatch score also reduced by giving the score by mutation rate, accepted mutation rate, also known as a point accepted mutation, that is a PAM. Then advanced format, that is a upgraded format of a PAM, is known as a BLOSSOM, block substitution matrix. So in this local sequence alignment, for increasing the matches, they use the mutation rate, that is accepted mutation rate. They vanish the gaps. It is somewhat similar to the BLAST algorithm, but the method is followed by the Smith-Waterman algorithm. Otherwise, it is similar to the BLAST. So the procedure, the same, blast.ncbi.nlm.nih. So it will open the page like this. Then we have to choose which type of blast we are going to do. You no need to worry about that. You can choose any tab. I have clicked there protein blast. That means protein to protein search. That is optional. You can also change later. Then you will get a page like this. By default, it is a blast page. Okay, I will explain. Here there are many tabs: blast N, blast P, blast X, T blast N, and so on. You no need to worry about that. For a protein conversion, simply click blast P. I will explain in detail later. For a protein to protein conversion, we have to select a blast P. Similarly, for the nucleotide, blast N. Now I have chosen BLAST P for the protein. Then third step. This is an important step. If you ignore this step, it will it will not change to the pairwise alignment. By default, it will search to the database. So be careful. You have to select this checkbox. Then only the layout will change. Now I have selected this checkbox. So you will get a layout like this. See, in the previous page, there is no another text box, only one, that is for a query. Here, there is a new text box that is to enter the subject sequence. Okay, so we have to careful to select this checkbox. Otherwise, it is basically a BLAST. You may confuse that what is the difference between the BLAST and this local element. Basically, BLAST is a local element. There is a meaning in the expansion. It's a basic local alignment search tool. So basically, it is a local alignment. The only thing is for the BLAST, they use a heuristic algorithm to compare a large sequences, not like a one or two sequences, billions and billions of sequences. So they use a heuristic approach. Basically, it is a local sequence alignment. Okay, I'll explain later. Then again, next step, we just click a BLAST. In the previous practicals, I have used the sequence same in this also. So the result will be somewhat similar. Again, we have to choose the alignment to see the alignments. Then here we can notice that in the global alignment, it is mentioned as a NW score, but here 1258 bits. Actually, the BLAST uses a bit score calculation. So in this, the score is calculated in ratio of a bits. Then the expectation value, the method used for search, that is compositional matrix. Then these are the things that is somewhat similar to the global sequence identities, positives and gaps. These two are advanced method that is used to screen the sequences based upon the bits. Bits means binary conversion, either match or mismatch. That is not a bit, 1 or 0. 0 means mismatch, 1 means match. That's a big concept. Okay. I think you are familiar with this. Then third tab, dot plot, also you might be familiar with this. See, you can notice that the in this plot it ends with the ends at the corner. Also in the right side ends at the corner. So it is homologous. Okay. Okay. Now we are going to discuss about the sequence search engine. The basis is sequence alignment. The only thing is, in the sequence search engine, be used to compare with a large number of sequences. That is also known as a data set or a database or something like a structure, function, or futures and anything. So the concept is somewhat similar. Like a pairwise alignment, we use the same approach to filter the database. For example, you can able to search from the NCBI database like GenBank, or from some other database, EMBL, or from PDB or DDBG or anything. So the data size will be very, very, very large. Okay. So in this sequence search, they use a word matching by filtering through a heuristic algorithm. That is first developed using a BLAST. That is, so it is very, very popular for a search engine. Even though there are many variants, still now BLAST is very, very popular, because in olden days, for the searching sequence, it is a very difficult task. If you give a query, you will get result after months or years, because the data size is very large. To reduce the task and increasing accuracy, BLAST have introduced a BLAST algorithm, also known as a heuristic approach, which uses a set of assumptions to filter a large database to match the sequence. Usually, the search is based upon these categories. Nucleotide query to nucleotide, that means query is nucleotide and the subject sequence is also nucleotide. Then second type, nucleotide to protein, then protein to nucleotide or protein to protein. Then this one is somewhat similar to the first approach. The only thing is, in this, before comparing with the protein, it initially translates the nucleotide. That is, first the nucleotide is translated to protein. Similarly, in the database, the nucleotide is translated to protein. Then it compare with the protein to protein. Okay. So, in both cases, it first translate the sequence, then compare with the translated sequences. Then, these two are especially for functional predictions and motif, domain prediction, and so on. Here, the protein sequence is compared against the PSSM, that is also known as the position specific scoring matrix. Here, the subject sequences are categorized based upon the families. Okay, it will categorize the set of sequences into motif, domain, profile, etc., and calculate the scoring to a matrix, and the protein sequence compare with the calculated scoring matrix. It is something different compared to the previous method. The thing is, first it will calculate a PSSM matrix, that is a position specific scoring matrix. Then instead of a sequence sequence is compared with the sequence structure or CDS, conserved domain regions, functions, etc., etc. That is especially for a functional study. Okay, these are the basic things we have to understand for a sequence search. Now, the practical session. First, we have to understand what are the programs which are used for the BLAST. As I have already explained, for searching nucleotide for the protein, then X represents the translation. Here, the query is initially translated, and the translated protein sequence is just against the subject sequence. So they use the term as a X. Then translation to translation, that's a T, translation plus X. X represents the translated form of the frames. That is 6 frames, also known as a ORF, open reading frames. 6 reading frames means for a codons, there must be at least 3 bases. That is, 3 bases will convert to a amino acid. So the reading frame will be different. For example, if you start from the first character, there will be possible it goes on 3. It begins with the 4. Again, it belongs to the first reading frame. So basically, there are 3 frames from the forward, 3 frames from the reverse. So totally there are 6 frames. So before comparing a nucleotide, it will convert into 6 frames, and the 6 frames, that is the 6 sequences, will compare against the 6 frames in the nucleated database, that is in the translated form. So it is known as the 6 frame translation against the 6 frame translation. Then another type, protein versus a nucleotide 6 frame translation. As I have already we have seen there, nucleotide 6 frame translation to protein. Similarly, okay, I will explain later. Okay, now the practical section. The procedure is same compared to the pairwise alignment and the local sequence alignment. First you have to open the browser, enter the URL blast.ncbi.nlm.nih.gov. Next, we have to choose the program. Okay, we can also change it later. Then we can notice that layout is somewhat similar to the local sequence alignment. The only thing is here the checkbox. Without selecting the checkbox, it will be a pairwise alignment. Here pairwise alignment means the second sequence, that is a subject sequence, is a database sequence. Okay. Here in this, we have to select the database which you are going to search. By default, it will be selected as a NR, that is a non-redundant protein database. The thing is, it will search basically it is a collection of data. The thing is, it is non-redundant. Okay. So it is known as a NR. There are many databases listed here. Here I have choose the protein database for a structure search. Actually, the protein database is for structure repository. So, for your understanding, I have choose sequence against a structure search. Okay. Then here the algorithm. Level will be very high compared to the default blast. This is strictly for the functional comparisons. The PSI that represents the position specific iterated. The thing is, the sequence are classified based upon the families. Similarly, second type, pattern. The concept is somewhat similar to the psi. The difference is the families are categorized and formed to a pattern. Pattern means like a sequence with a set of rules. So basically, psi and phi blast N, blast P, and so on. Either you can select here itself, or we can change it later by clicking these tabs. Okay, this is a first tab, that is a description about your sequence and so on. Here, to select the highly matching sequence, we have to use the here right side scores, e-value, percent of identity, and so on. Here there is also option to search through the GenPept, that is a translated form of GenBank, or to display in a graphics or calculate a distance matrix and so on. This is new features added in this blast. It is not available. To screen your search sheets, we have to first find out the of identity. Here 100%. So it is exactly same. So your query sequence is a crystal structure of the PD is a domain, something else that is deposited in the protein data bank, which is belongs to the species Homo sapiens. Exactly, it has matched. If you are familiar with the BLAST algorithm, then you can choose this one: maximum score, total score, or e-value. Okay, this is strictly for a algorithm, that is for a developers. Those who have it will be easy to understand.
Sorry for the inconvenience. Due to some technical issues, we will start shortly.
Sorry for the interruption. Okay. So for choosing the matching sequence, we have to see the query coverage, then percent of identity, then directly you can link to the database. Here I also use the PDB. So the link is redirect to the PDB. Then this is accession number of the PDB. The first four letters represent the PDB ID. Then last letter is the chain. So A represents the A chain. Here is also B. That means the B chain. Because the protein may also consist of many chains. Okay. So we can easily interpret that it is matching with the first chain. That's the A chain. Then okay. If you want to see the alignment, you can click this alignment tab, or to find the taxonomical classification, you can also choose the fourth one. Now we are going to search for a sequence, structure. So I have ignored these steps. So third step, we have to click the link. So it redirects to this page. This is a default GenBank file format. The PDB that is matching with the domain, it is linked in the right side. So if you click this link, it will redirect to the PDB, that is a protein data bank. Okay. Okay. Next practical session is a multiple sequence alignment and phylogeny. This is very, very important in sequence alignment, because previously we have just using the alignment for either to compare a sequence or to search a sequence. But this multiple sequence especially used to classify a sequence or to find out the families, that's to find out the evolutionary relationship. So for this evolutionary relationship, first we do a multiple sequence alignment. Then from the multiple sequence alignment, we then predict the evolutionary relationship. The evolutionary relationships are usually represented by graphs like a trees. Okay. The evolutionary relationship represented by the trees, that is in a 2D graph, is simply known as a dendrogram. Dendro is a computational term, simply means the graph with the hierarchical representation. Okay. For bioinformatics or computational biology, we use the term phylogeny. Basically, both are same term. The thing is, the dendrogram is a technical term used for the computers, but phylogeny is especially used for the phylogenetics. Phylum means classification of genetics, genes. Okay, we shall see what are the basic things we have to know. Then in the dendrogram, there are many types of representations, like unrooted, rooted. These are the broad classification. A tree is simply categorized into unrooted and rooted. See, this is an example. Here in this, it is unrooted. Unrooted means there is no common root. Okay. The branches are splitted without no common nodes. So it is known as unrooted. Then remaining thing other than unrooted are known as rooted. Here it starts with a common node. Here is also common node, and so on. So these types are broadly categorized into a rooted. Then strictly classifying, the rooted trees are also represented as a slanted, rectangular, and circular. Okay. This is a slanted. The lines are in slanting. Okay. So it is a slanting. So slanted chronogram or slanted dendrogram, then slanted phylum, anything else. Then this in the rectangular form. So it is known as a rectangular phylogeny. Then it is the curved form. So it is known as the curved phylogram. Then circular form. It occupies a space according to a circle. So it is known as a circular phylogram. Okay, you might be confused that there are many terms like a dendrogram. There is also another one word, cladogram, chronogram, phylogram. The thing is, that is a cladogram. Clads means the branches with no character changes. It is known as a cladogram. Actually, in genetical study, we don't use the clads. Here, it is one example. Here, if you see, it is properly arranged, but the branches have no meaning. The length are uniform. There is no variations. Okay. So it is known as the cladogram without variations. So it is known as the cladogram. Okay, even though it seems like this one rectangular phylogram or rectangular chronogram or something like, but the difference is the branches are not equally calculated. So it is a cladogram without differences, no character change. Then chronogram. The chronogram means the length are calculated properly and the length are arranged according to the proportion to time. For example, in this graph, you see even though the branches are calculated, it is arranged to the corner. See, the length are touching the corner. Here also the each and every length are touching the corner. Here is also. Okay. These types are known as the chronogram. Even for example, these two are basically same. The only thing is here the length is small, but you can notice that here the length is large. The remaining length are adjusted according to the maximum length. That is, for example, this is the maximum length, so it is arranged accordingly. So it is known as the chronogram. Okay, but phylogram is strictly calculated branch length. That is the difference between the phylogram and chronogram. Basically, both are same. The only thing is the length of change is proportion to the time. In bioinformatics, we choose this phylogram. So only generally these studies are known as the phylogenetic trees. Strictly for the phylum. Phylum means the classification. Okay. Then this is one type of representation, basically a rectangular phylogram. The only thing is in some graph they mention the length of variation. This figure is very small, so I think you cannot able to see that. The value is something 0.7 something like this. The length is represented in each branch and sub-branch like the nodes. Okay. This is one type of representation. Usually depend upon the graph, they will mention the distances. Because if you mention the distance and labels, the graph will be very large. You cannot, we cannot able to display in the slides. Then, for a phylogenetic trees, this is the order we have to follow. First, you have to decide whether you are going to display in a rooted or unrooted. Now, if you decided to display in the rooted, the next step have to decide whether you are going to categorize into cladogram, chronogram, and phylogram. Then, if you decided either cladogram or chronogram or something else, then the format to display slanted. The drawback is in the slanted format we can able to display the adjusted distance only. So most of them don't prefer this slanted. So we cannot able to use for these phylograms. Then rectangular, curved, or circular. These are strictly used for the phylograms. Okay. This is a procedure to construct a phylogenetic tree. So you must not simply construct a phylogenetic tree. You have to decide which type of diagram you have to going to generate. This is the important thing. Usually for the bioinformatics or computational biology purpose, we prefer this rectangular phylogram. Okay. Then depend upon the space of your documentation, we can also choose this one with the distance. These are the basics of a phylogenetic tree construction. Okay. Now we can enter into a practical session. Then Cobalt. This is a most popular multiple sequence alignment tool. It is also an improved tool. In olden days, cluster W is very popular for the multiple sequence alignment. But in this Cobalt, they used advanced strategy to improve the accuracy at a sequence level, functional level, and domains, etc. The concept they implement is a conserved domain. So it is very popular because sequence alignment is not only for comparing a sequence. The main aim is to compare a function, because human sequence may also match with a human or Homo sapien or like a Drosophila or like this other species also. The only thing we have to understand is whether the functions are same, because if you are predicting a sequence to design a drug or to design a vaccine, etc., we cannot able to trial with the same species. Like for example, we cannot able to trial with a human. So we choose a closely related or evolutionary related species like mice, rabbit, etc. So it must be also evolutionary related. For such kind of purpose, the domain is very, very important. We have to prefer. So it is very popular for the multiple sequence alignment. Even during this corona, there is a COVID pandemic, most of them choose evolutionary study to trial the drug. Okay. This is especially based upon the domain search. Then the procedure is somewhat similar to the blast. Only thing is we have to choose the multiple sequence alignment. Then here either you can enter the sequence in this text box or upload the sequence if you have downloaded from the database or stored in a computer. You can directly upload this. Then click the align button. So this is the result. Here I have hidden some of the section to display everything. Okay, actually it will show here a graph that is a conserved domain region. I have hidden this section. Here also one section I have hidden. Here the important thing, we have to find how it is matching with all sequences. Usually the characters that is matching is represented by symbols or colors. In olden version, it is represented by the symbols. But in this new version, to reduce the space, they use the colors. See here the letters, some are displaying in colors, red region, sub blue region, etc. Here there is a number that is represented by a square brackets. That means there are some mismatching regions that is beyond 4-letter characters or within 4-letter characters. If it is within 3, they will simply display. Here you can notice that P or G, only 3 characters. So they have displayed. If more than 3, to reduce the gap space, they simply represent it by square symbol with the number of characters. Here it is 15, like that. If you want to display a complete sequences, we have to select this full. By default, it will show compact mode. Compact means compressed form. Okay. To display the full alignment, you have to choose the full mode. Then, okay. Then for constructing the phylogenetic tree, we have to click the link phylogenetic tree. Okay. This is the phylogenetic tree. Here it is in the format phylogram. Okay, you can choose tree method. There are many types of tree: NJ plot, MS format, FME, that is a fast minimum evaluation, and many types. By default, it will show fast minimum evaluation. You can also choose the distance calculation. By default, it's a Grishin. It is also popular for distance calculation. Then you can also able to change the representation. Here I will choose the sequence title, so it is displayed like this. You can also change to access a number or id or simply without label. That is depend upon your choice. Okay, this is a standard format. Most of them prefer to phylogenetic tree constructions. You can also choose any other format as I've already explained in here. Every options are available. Okay, that's all about the multiple sequence alignment using the cobalt. Then the next similar practical, that is a cluster omega. Olden version, it is also known by cluster W, cluster V, cluster X. The cluster omega, also known as a cluster O, it is a popular tool. They use a HMM profile to profile techniques. Here they use a hidden Markov model, that is an artificial intelligent approach that used to compare with a set of sequences that is similar to the functions. So it is known by the HMM profile to profile. HMM means hidden Markov model. So instead of basically comparing with the sequences, they used the hidden Markov model as a data set, and based upon this HMM, that is a hidden Markov model, it compare with the query sequence. So it is very advanced compared to the cluster X or cluster V or cluster W. Also it is very fast. Okay, now the practical session. First open the browser and enter the URL like this: eba.ac.uk/ important thing, in the URL the T is capital letter. Some of them use the habit of simply entering the small letter. If you enter simply small letter, sometimes it will show it is the URL not available. So you must be careful that in this URL the T is a capital letter. Tools MSA cluster O. Okay, then the next step, you have to choose what type of sequence you are going to enter. Here I have chosen the protein for the protein sequence. Then we have to enter the sequence either in FASTA format or simply upload a FASTA file, or you can also try using this option, using use the example sequence. If you click, is automatically paste in the text box. You can try using this option also. Then standard format to display the multiple sequence alignment. By default, it is a cluster W format. The olden flare, that is of a cluster W. The cluster W is very popular. The formats also followed by many multiple sequence alignment tools. So by default, cluster W is used to display in the multiple sequence format. Then finally, click the submit button. Okay. Here, by default, it will show the alignment like this. It will be slightly different compared to the blast multiple sequence element, that is cobalt. The thing is, at the end of the row, here I use the three sequence. At the end of the row, they use a symbol representation to show the match, mismatch, etc. The star represents the identity. Then double dot, that is a column, that represents the maximum possible chances of matches. Then dot represent the minimum possible chances of a matches. If there is a gap, so the probability is very less. That is evolutionary less related. Okay, these are less related. Because this cluster omega is based upon the guided tree, for a profile to profile, strictly based upon the functional study. So even though if it's a mismatch, it will search for the evolution relationship. For example, if a sequence is belong to the same family, like a grandparent, parent, child, grandchild, grandchild, and so on. These are all belong to the same family. So it is it can be accepted as an accepted mutation. But in this case, there is no symbol. That means it is not accepted as a mutation rate. Then there are many options. You can try yourself. For a phylogenetic tree construction, we have to choose this one, the fourth tab, phylogenetic tree. The only drawback in this cluster omega is the phylogenetic tree construction is very poor. See here in this simply there is a three lines. We can't able to differentiate. That is one of the drawbacks in this cluster omega phylogenetic tree construction. Even also you can able to choose other tools like phylogenetic tree construction. I will explain. Okay. The important thing, while displaying the phylogenetic tree, there are two options. That is a cladogram or real. Simply, it is a cladogram. To find out the difference, we have to select this option real. Okay, now I am selecting this option. See, you can able to find out the difference. Actually, this is also very poor. Based upon the distance, the graph must be like this. The root must begin from here. Okay. So if we want to analyze this tree with some other phylogenetic tree software, you can simply copy this data. This is a phylogenetic tree format, also known as the ASN format. Simply copy this content and save in a text editor to phylogeny format. For example, some file name .phy or ph, and we can open in any one of the phylogenetic tree. You can display as you wish. But in this cluster omega, only two options are available. The tree is very poor for displaying. This is one of the drawbacks in this cluster omega. Otherwise, most of the features are good compared to the cobalt. Okay. Then the last one. Actually, the most of the tools are very limited for this physiochemical or biophysiochemical properties prediction. So I have selectively only mentioned some of the popular tools. Okay, this physiochemical properties prediction is simply like predicting properties from a sequence. So for a nucleotide properties, we can able to predict residue length, composition, frequency, complement, reverse complement, translation, transcription, reverse translation, ORF, and so on. One of the popular tool used for this is SMS. Then emboss is also very popular, but now it is depreciated. So few popular programs only included in EBI. Actually, this was developed by EBI. They now host only few programs which are popular from this package. That is the European Molecular Biology Open Software Suite. During 2000, it is very popular. At the time, there are no such tools are available. Then later, the SMS became popular because most of the features are published by this SMS. Okay. The another one feature is it is very fast compared to this emboss. It is a Java-based tool. Then the last one, protein properties prediction. There are many properties. I have mentioned one of the similar like length, composition, frequency, molecular weight, atomic composition, and so on. There are many properties. Even we can able to find out the structures, predict the sequence to structures or many things. But for the nucleotide, the tools are very limited. The popular tool for the protein properties are protpram, that is belongs to the XPASI. Then AA-prop. The features are more or less similar. These tools, these two tools are from my server. This tool I have hosted in my server. Okay, we shall enter into practice session. This is my server. The features are same included in this tool. So we have to enter the URL biogem.org/sms2. This new version, the older version is sms/. Or you can also choose the first one. This one is a mirror site. Here lot of programs are available. I have mentioned only one. You can try yourself. Here I have selected for molecular weight calculation. If you simply click, it will open a layout. By default, it will show one example, or you can clear it and enter your own sequence. I have entered my sequence. Then just click the submit. Then it will open the result in a new window like this. It will open in a new pop-up window. It is very fast. The thing is, it is a JavaScript based program, so the compilation will be very fast. The results will show instantly. You can use molecular weight, DNA statistics to find how many number of bases are in the sequence and so on. Then multiple reverse translation, translation. These are for the proteins, ORF finder, and so on. There are many programs. I have highlighted only few. If you scroll down, there are many programs. These are all used for the nucleotide or protein property. Basically for the nucleotide, you can also use for protein properties like a protein gravy, isolatory point, molecular weight, and so on. This is a very simple tool. Then and the last one, this is for a amino acid sequence properties prediction. So this is one of the tool from my server. This is a short URL or this is a full URL. You can choose any one. It will direct to this URL. Then the layout is very simple. You can either try with a sample sequence or enter a sequence. The important thing, you have to enter raw sequence. As I've already explained in the beginning, there are two types of file formats usually used for a sequence analysis. Here, I have choose the first format, that's a raw file. Raw file format means just a sequence. Okay. Then click the predict button. Here, there are a lot of properties are predicted. I have highlighted only few. So you can see the features by scrolling down. Okay. Okay, we have came to end of the session. Sir.
Hello, sir.
Okay.
Hello, sir.
Yes. Huh? Okay.
So there are few queries.
Okay.
The first question was, what is the use of machine learning in bioinformatics?
Good question. Actually, there are a lot of problems in predictions using a computation tool. Actually, the computation tools are simply known as a GIGO, garbage in, garbage out. What you give is what you get. The thing is, if I am a developer, I design a tool to predict functions or something else. Then if I give if I mention some mistakes in my algorithm, the result will be also according to that. So to rectify these kinds of problem, the artificial intelligence program like a machine learnings are introduced that decides by the program itself. So the level of accuracy is very high. Also, another one important feature is instead of entering the data, we can combine the data set, and the data set is automatically predicted by the machine itself. So only we may known as a machine learning. The machine automatically decides what function it will be predicted. Hello.
So next question was
Ah.
how to compare amino acid sequence similarity.
Basically, the comparison is done by three basic rules: match, mismatch, and gap. We all expect only matches, but while we compare either with a protein or nucleotide, there are some of the common problems. That is, DNA or RNA or amino acids all consist of same characters. That is, the characters in the alphabets are same. So the modern algorithms, they use a common character set, like for example, biological sequence characters or biological sequence alphabet that consists of all characters. Either it is a DNA or RNA or protein, it simply finds the match, and the sequence will be matching with the functional coding regions, like evolution related species. So there will be the problem is reduced by sequence to function. Instead of a sequence to sequence, the prediction will be sequence to function. Did you understand?
So the next question was what are the primary and secondary databases in bioinformatics?
There are many databases, many. But for your workshop purpose, I have only mentioned only few. Even each and every year, many researchers are developing database. Among them, the most popular databases are for the sequence NCBI, then for structure PDB. Also, another important thing is nowadays, even though we are releasing a database, most of the data contents from the other database also merged, that is combined. So based upon this, we categorize as a primary and secondary. The primary means the data which are deposited from the experimental, that experimental results are stored in the database. So it is simply known as a primary. But in case of a secondary, there are many tools are now available to predict the functions. So using this primary data, they predict the functions and deposit as another database. So it is not the secondary database. It is a common type of classification, but nowadays most of the researchers are involved in functional study. So another type of database also released, like composite. They combine the primary data as well as the secondary data. Then based upon the primary and secondary data, they predict other data using some tools or like algorithms, etc. But based upon the molecular classification, we can classify a database as a sequence, structure, and function, depending upon your choice. You can simply classify.
So next question was which multiple sequence alignment tool is better?
It is a big question. Actually, bioinformatics started with a set of assumptions. So we cannot give 100% accuracy. Up to till date, there is no tools promise that we can give 100% accuracy, because even though I am also a programmer, I used to design a tool based upon set of rules only. Okay, so the rules, for example, I have a standard rule: DNA consists of some set of alphabets, or RNA consists of set of alphabets, or DNA consists of certain molecular weights or atoms and so on. It is a standard rule. But nowadays there are many types of molecules are discovered by scientists, like a mutated genes, or previously scientists have proved that there are only 20 amino acids, but now they have found 2 amino acids, so they say as a 22 amino acids. Also they found some variants, so they say as a 25 amino acids. Now researchers are confused because English alphabets consist of only 26 character. What if it is there are more amino acids if we find beyond 26? So we cannot represent by a standard English alphabet. So it is a big confusion. Like that, there is no well designed protocol in tool development as well as some other for a database. They cannot able to form a defined data. So we can able to confirm whether the data is similar or not depend upon our knowledge only. The computer will give result as you give, it will give the result. That is a garbage in, garbage out. What you give is what you get. But nowadays there are many artificial intelligence programs are developed which gives more accuracy. But from my experience, for sequence similarity, you can choose a BLAST. Then also for the multiple sequence alignment, you can choose a Cobalt. They are not only for not only based upon the set of rules, they are also function dependent. Okay. The another one problem is nowadays there are free server and paid server. For academic purpose, we can only use the free servers, but there are many paid servers, there are commercial tools which gives more accuracy. Thus, such kind of tools are accessible from the like private companies or R&Ds.
So next question was, what is the difference between local alignment and global alignment?
It is very simple. Basically, for the sequence alignment, we use 3 rules: either match or mismatch or gap penalty. We expect only match, but there may be possibilities of mismatches. Without using a mismatch or gap penalty, we cannot match a sequence. So we use some penalties and some mutation scores to align the sequence. So in the global sequence alignment, they use 3 scores. They use a match, mismatch, and gap penalty. But in the local sequence alignment, strictly we use a maximum possible of matches. So in the developers strictly reduce the gaps. For example, they use 1 or 2 gaps only. If there is more number of gaps, they use a penalty score very high. So the gaps are totally vanished. So if a gap is reduced, then it will be local. Only the exact matches or more number of matches. The thing is, the mismatches and gaps are reduced in the local sequence alignment. Strictly gaps are reduced. So only we say as a global as a entire matching, local as a limited match.
The next question is, what is BLASTX and why it is used for?
BLAST is basically basic local sequence alignment tool. The concept was inspired after the development of local sequence alignment. But the big challenge while developing the tool is now the database size is very, very large. We cannot able to imagine. Even a whole genome sequence, one sequence size may be 10 GB. So the database size is very, very, very large, trillions and trillions of size. So if you go for searching a query sequence against a database, you cannot get to result in your life. The computing process is very, very, very large. So to reduce the task, BLAST has implemented algorithm known as a BLAST algorithm. Also, it is known by heuristic algorithm. The heuristic algorithm means in the algorithm which is developed using set of rules. They used many assumptions to reduce the database size. For example, instead of directly compare with the sequence, because the pairwise alignment compare each and every characters, but in case of a blast, they first filter the data content by a families. Then the families are then filtered by the profiles and domains. So if it is a large sequence, for example, 1 lakh of sequence is filtered to 10,000. Then the while we use a domain search, then the 10,000 will filter to the 100. Then from the 100 we use the local alignment. So only the term is basic local alignment. Then they use a local alignment approach. Then the local alignment approach, they use a bit score. Actually, the bit score is for a binaries 1 or 2. They take only 2 decisions, either match or mismatch. So while using the decision, if there are more number of mismatches, they just ignore the sequence. So the 100 reduces to 10. Then the task is between the 1 to 10. Then they use basic scoring like a match and mismatch and gap penalty. If there are more number of gap penalties, then they go for next approach, that is accepted mutation. So if you go for accepted mutation, the task reduces to 3. Just I am giving an example. Then you can easily find out the closely similar sequence. This is the approach used by the BLAST. So only it is a very popular search engine for a sequence search. Even for next generation sequencing, they use this BLAST, because next generation sequencing is based upon the big data technology. The data is not like as we seen in a practical, the sequence will be very short, not like that. Whole genome sequence will be very large. So nowadays they are using the big data analysis. The server is very fast and the algorithmic approach is not like a matching and mismatching. Instead of matching a character, they use a word. For example, there is a letter A to A, there may be more number of combinations. If I use the word Ashok, then there will be limited combination. Like that, they use a word matches. This is a secret behind the BLAST.
So the last question is, while working with explicit translate tool from nucleotide sequence, why do we have to choose the longest highlighted chain in different frames?
Actually, one of the big problem is sequence to functional prediction, because there is a strange feature that a nucleotide has the possibilities of translating more number of proteins. For example, codon region consists of 3 bases. So while we reading from forward direction, there will be 3 possible chances. For example, 1, 2, 3, 2, 3, 4, and 3, 4, 5. Then if you go for 6, then it will repeating. So in forward there is a 3 chances. Similarly in the reverse there is 3 chances. So totally 6 frames are there. Then if I go for in the translation, you may also get a 6 protein sequence, but practically there is only possibilities of 1 protein. So for this, we cannot able to easily determine whether one sequence is matching. So we have to find out all possible chances. Among the all possible chances, we have to decide one. So based upon this, for example, there is a 6 possible chances. Then from the 6 possible chances, we can determine which is highly scoring. So only use this translation approach.
Thank you, sir. Next, I welcome Ms. Anjusha P. for the vote of thanks.
Thank you.
Thank you, sir. Thank you for enlightening us with an informative session on sequence analysis. It's sure that the guidance that you provided during the workshop would be very helpful for us to develop our skill regarding sequence analysis. We also like to express our gratitude towards Dr. T. Ashok Kumar for taking out time from his busy schedule and lending a friendly and supportive hands for us. We thank all the participants and faculties for being a part of this workshop. Once again, thank you, sir. Thank you all.
Thank you, Noorul.
This comes to an end to our bioinformatics workshop. Thank you for everyone who participated in this program. Thank you.